the human has co-evolved with trillions of bacteria within our body. The growing evidence or the growing knowledge on this association with the microbes and the human body indicates that they are doing diverse functions, and this knowledge can be utilized further to improve this association in a way that human can adapt to better way in the changing environment in the planet. Darwin did the Beagle voyage, as you are aware of, in the year 1830. He did it for 5 years. He collected information on diverse life forms found in the planet. Although he didn't visit India, but he had good knowledge on the diversity of life forms in India. And finally, he came up with the great theory of evolution, as we all know in his book, The Origin of Species by Natural Selection. The microbiologists were not very happy with the theory, and they call it sterile theory of origin of species because Darwin didn't mention about microbes in the theory. And the point that he was contemporary with Louis Pasteur. They were at the same time, and he was aware of the work of Louis Pasteur, and also in the just last decade, Antony von Leeuwenhoek and then Robert Hooke, they had already described about microbes, and probably at that time microbiology was in very infancy, and there was not well studied. It was not considered a well-studied discipline at that moment, and or maybe he didn't want to step in the whole mess of microbiology, or maybe his theory already covers all the aspects of life forms. And it can be found from the indication that he said that if any part of any organism can be proved that it supports the another organism and exclusively for the benefit of the other organism, then his hypothesis will be proved wrong. And of course, we understand that natural selection cannot support this way, one way, associative. In this planet, we have diverse life forms, as we know that microbes outnumber in terms of total number of species. We have more than 9 million different species of microbes. Taken into account, if you take all the species of animal and plant, still microbes are higher. And all these higher plants or animals, they are very complex form of life, and they have evolved from very simple single-cellular organism, as you have already seen in the last few presentations. And the major thing here is that these complex life forms, they have evolved from very simple form in a series of sequential transitions. And those transitions were either for nutrients or maybe for sources of energy. And if you look at the last 40 years of history of the modern mankind, you'll find that in the first 30 years, we have already learned how to domesticate animals, which led to subsequent agriculture. And also we have learned how to use the tools. And if you look at in the last 100 years only alone, we have learned how to fly, we have learned how to build computers, we have learned how to send rockets, also use thermal energy. So if you look on this curve, you'll see that within this period, the curve is moving towards vertical. It's almost going vertical. So, and if you want to know further about the technologies, those will be dominating our near future, you are most welcome to visit this website. This Futurism is a very nice website. Particularly students, you should follow this one. You can have a link so that all the new things comes to your mailbox. Here, Stephen Hawking has the opinion that we have only 100 years left in this earth. We have to find some alternative planet for our future habitation. Otherwise, the mankind may go extinct or something may happen. And you know the person, Elon Musk. He's the CEO of Chief Executive Officer of the company called SpaceX, who are working on the various things about aspects of space. he wrote a very nice article in the latest New Space journal. He says that in the planet at the moment, the mankind has two options. One, either you stay here, wait for your fate, or you can find another habitation somewhere else in some other planets. And he suggests that the Mars will be our next habitation, probably, because it is most suitable. And he has justified in a very nice way. You can just go through his article. And during this process of modern technological evolutions, we have modified the environment in the Earth in such a way that we have not only we have modified our atmosphere, but we have also modified the space around the planet in such a way that now it is said, maybe you are aware that low magnetic radiations have created some kind of shield around the Earth, and it is protecting from space radiations. So whatever may be the case, one thing is clear, that in the near future, we will have less physical activity, we will have more mental stress, and we have to face the climatic stresses in various forms. But the question is, human being cannot evolve that fast. If you look at the last 4 billion years of history of evolution, human cannot evolve that fast. So what is the option? So microbiologist has the opinion that there may be one hidden design inside the superorganisms or the complex organisms. And they gave forward the concept of holobiont. According to this theory, holobiont as an organism is constituted of two parts. One is host, another is the component of the symbionts. And symbionts, and these two combinedly called holobiont, and that should be taken as a unit of selection as per Darwin's theory of evolution. And symbionts, you have already good knowledge on this, I think. In the last talk, sir mentioned about core and transient one. If you take the core symbionts and the host, the way they are transmitted to the next generation, I think they do it in such a fidelity that it fits into the Darwinian theory of evolution. And in the host, human being, we know we have around 23,000 genes. Okay, and in the symbionts, together at any average person, like the symbionts can contribute to around 3.3 million genes, and these genes are now the target of modern biologists. So they think that this magnitude of genes can simply probably, or if you can manipulate somehow, or if you understand properly their functions, the human being probably will survive the imminent threats that will be coming in the near future. I give few examples of close association. You already know about rhizobia. It's a bacteria that forms nodules in the leguminous plants. Leguminous plants, you know, the plants with like peas, who's formed, with that kind of trifoliate leaves. So in the roots, these bacteria form nodules, and they form very close association, and it fixes nitrogen for the plant. The plant in nitrogen deficient soil cannot survive with this bacteria. Again, maybe you have heard of Wolbachia, is a bacteria that lives in the cytoplasm of many insects and also some nematodes. And in many insects and nematodes, they are so tightly associated that the organism cannot survive without the presence of this bacteria. And this knowledge on this association has been utilized to control some vector-borne diseases, including dengue. Probably you have heard that in China, one company have introduced a type of Aedes mosquitoes to control the natural population of your Aedes mosquitoes that carries your dengue virus. And this bacteria doesn't allow the dengue virus to replicate within. And what is the concept here? That if male crosses with female without getting the bacteria, their first generation is like lethal, they cannot multiply. And then if you use both male and female containing the particular bacteria, the next generation will carry, all of them will carry the bacteria. Now, in this case, if the female contains in the cytoplasm the particular bacteria, and even male doesn't contain, all generation will be, next generation, all the members will be like this one, containing Ulvasia, and the disease will be controlled. The mechanism is that because they are found in the cytoplasm, and then so it will be going through the female, because the sperm doesn't contain your this one cytoplasm. And interestingly, another one fact I want to share with you. You know about mitochondria, it is endosymbiont, Sir has already described. And rhizobia, Ulvasia, anybody can guess who are they? Like in microbiologists? All of them are alpha proteobacteria. So during the process of evolution, this particular lineage, probably they have conserved their way of association with higher organisms. I give another example. You know, mealybug, maybe in this season, you will find particularly in citrus plants, and also if you have brinjal, solanaceous plants in your garden, particularly in this season, rainy season, you will find this insect, this kind of cottony growth. Okay, this is in tomato plant. And what happens here, in this insect, you find 3 levels of association. One is, these are the cells of your particular mealybug, and within these cells, you will find another bacteria, Trembalia, and within these cells, you will see the green colors. There's another bacteria, okay? And the amino acids tryptophan and phenylalanine cannot be synthesized by alone of these 3 organisms, but they produce this tryptophan and phenylalanine in a shared pathway mechanism. So this association is very tight. You can imagine that they cannot survive without each other. I give one example how the gut microbiota can influence the speciation. You know, for probably zoologists will know that for speciation of any organism, the first step is your mating preference. And if you grow fruit flies, Drosophila melanogaster, 2 groups, one group in a media containing starch, another with molasses, they don't prefer to mate with each other. Okay? And then if you treat with tetracycline, they mate. So you know better, tetracycline, antibiotics, what they do. They kills the bacteria inside and they don't mate. Now, if you isolate a particular gut bacteria from this starch growing fly, and then if you feed it here in the antibiotic treated this one, they again don't mate. So their mating preference again comes back due to that particular bacteria. And now we know that this bacteria synthesizes some kind of sex pheromone which the other doesn't like. Similar example came in the year 13, where Brucker and Borsin, they have shown that in wasp, is called Nasonia, they have subpopulations with some genetic variations, and if you cross them, the first generation that comes, they are lethal, they cannot survive. And then if you treat them with antibiotic, they survive. Again, if you do similar kind of microbial isolation from this, again, if you feed the antibiotic treated flies, and then again it becomes lethal. So it is very clear from this one that the microbes are very much essential in this particular genetic makeup, and then if that makeup and the association of the particular genotype and the particular group of microbes, if it is distorted, they cannot survive. And during the course of evolution, I would like to share with you this information that we have acquired many genes from different organisms. So one example I want to give, our microbiome also acquires genes from other organisms from the environment. The agar that is used to solidify our culture media, both in plant tissue culture or your microbial media, the agar is used. And if you take this agar, you cannot digest it. It is polysaccharide, it's carbohydrate. It is extracted from seaweed called agar-agar, and you cannot digest. For digestion, you need a particular group of glucosyl hydrolase family genes. It's called agarase. They synthesize agarase enzymes. And this enzyme is found in some bacteria in the marine environment. And recently it has been shown that the Japanese population, they have these genes in their gut bacteria. And those bacteria, actually the gut bacteria, they have acquired these genes from the marine microbes. So they are giving some kind of fitness to the host to digest this agar. Because the Japanese people, they take sushi wrapped with your seaweed, and also they take seaweed in different preparations. And even in our genome, we have 30%, 37% genes lineage you will find there from bacteria. And 120, more than 20, I think it's around 40 genes, those are not found even in primates, our nearest ancestor. You will not find in them. Those genes are acquired from bacteria. Gut bacteria, they influence many ways our body. You know about it. My students, they have already presented many aspects of this. What I want to show here that there is one experiment which shows that if you take the gut microbiome from an obese person and from a lean person, and then if you transplant that into like mice who like lack of doesn't contain the intestinal microbes, they are clean. Like it's called germ-free mice. Okay, and if you you make 3 groups. Okay, one group you transplant the microbiome from the lean one, another group from the obese one, and in another group you take the particular dominant bacteria here, it's called Christianelaceae. If you supplement with this particular bacteria, again, these mice is lean. Like one will be obese, from lean is again lean, and the obese one supplemented with the particular bacteria can make the mice again lean. So it indicates that the gut bacteria, particular group of bacteria, like they have some role in obesity. Maybe obesity, this is not the single factor. There will be multiple factors. This is an additional thing. And the overall impression now at the moment is that lean mothers are more likely to have lean offsprings, and then obese mothers more likely to have obese offsprings. The gut, human gut, is such a complex organ of the body that anatomists, they say that it is only the second to brain. The neural network is so complex that it connects to brain through vagus nerve to various regions of your brain. And in those regions, those are responsible for emotion, stress, anxiety, hypertension, so many things. And microbes in the gut, they are known to produce different compounds to interact with the brain with some molecules like called GABA, and then you have serotonin, dopamine, and they can have immense influence on our mental health. One classical example that I want to give, the group of scientists, Bravo et al. From University College Cork, they have shown that treating mice with particular bacteria, Lactobacillus rhamnosus, can motivate the mice. So how do you test that? The people who work with mice, maybe they're knowing that there's a test called forced swimming test. You put a mice inside water taken in a glass, and the mice, particular mouse will try to swim and come out of the container. Okay. Now, if it is depressed, it is not highly motivated, after some time it will stop swimming and waiting for the fate. Okay. And then, if it is highly motivated, it will keep swimming for a longer time. That is the test, normal test for your antidepressant drugs, like people might be knowing. So, after the treatment with particular bacteria, they found that it has, positive motivational effect. And many studies have shown, like particularly in a mice model, that different group of microbes, they are responsible for mental well-being, and they're thinking about behavioral transplant in human being in future. Toxoplasma gondii. This is not a bacteria. This is actually protozoa. Okay, and why I am being here, because it is not a gut bacteria as well. So it is found in half of the population, anywhere, maybe half of us carrying this particular organism, and we act as a carrier, and sometimes it shows some diseases conditions, or otherwise it will be in hibernation. And it can influence the fear factor of human being. It can affect your risk-taking behavior. There was one very good study carried out on the soldiers of Czech Republic. What they have done, they have scanned this number of soldiers for the presence of particular organism, and then they have observed their history in the subsequent years, like their behavior for several years. And they don't know about this study. I mean, they're not aware of the Toxoplasma thing. And they have collected information from the military police. And they found that presence of this particular organism increases the risk of traffic accidents. And what they found after this study, that this particular organism hibernates in a place called amygdala in the brain. And this part is known to control our fear. So once it is there, so it is disturbing that particular signaling thing, and you are more prone to road accidents. I give another example, Helicobacter pylori. This is one organism that maybe you know very well. So in 2005, these two gentlemen, they got Nobel Prize for discovering that this bacteria is found in our stomach, and sometimes they can cause your ulcer, and that can be cured with subsequent treatment with the antibiotics. So they got Nobel Prize. So this is one story. The another story I'm going to tell you, that this bacteria is so tightly associated with mankind that you can find the migration history of the human being based on their genotypes. So in African population, you find 3 different types. In Europe, you find 2. Asian population has different one. And then if you nowadays the population genetics, when they study the genetics of the population, they also now use this type of the Helicobacter pylori to whether substantiate their data. And one interesting thing is like this mummy, maybe some of you know, is called Odzi. It was found in the Alps mountain on a place called Ordzlel, so it came from that word. So they gave the name Odzi. It's a naturally mummified body of human being. And recently they have identified a type of Helicobacter pylori in its gut that is found in the African population. And it fits very nicely that you know that European population, the modern European population, they migrated from Africa. So 5 years ago, this person had that type of Helicobacter pylori in their gut. Apart from this gut bacteria, we know that they can modify the drugs we take. They can either activate a drug you are taking, or they can inactivate, like the cardiovascular drug that you take, deoxygenin, can be deactivated by gut bacteria. And also they can be made toxic, like the normal paracetamol. In some people, they may be hepatotoxic if it is converted to a particular group of bacteria. Now the question is, when we test drugs, we test like drug and human being, cause and effect. Now, with this background, maybe we should think about consulting the gut microbiome, what they say about the drug that you are designing. Similarly, what I want to put this slide here, particularly to show that this microbiome has not been explored for its total capability to help us with these kind of situations. I tried to find out many literature, but I didn't find proper literature on this. But what we find that these bacteria, they can reduce heavy metals in the laboratory like studies. But because they are our commensal bacteria present in our gut, whether they can be tamed in such a way that they help us in detoxification of the heavy metals, or the pesticides that we use in the agricultural fields or that come in our food, can we design, or can you think about designing the chemicals in such a way that the microbes will modify, or will modify the microbes in such a way that they modify these pesticides in such a way that they cannot harm the body. Okay, they are excreted or some way. So this is another aspect. And one more thing about climate change. We know that with the climate change, there may be several aspects apart from the temperature. But I tried to find out there could be any link, but I didn't find. But there was a study in Switzerland. They've shown that with the cold thing, the gut microbiome changes to burn more adipose tissues. So if it happens with the cold, maybe in the higher temperature also, you might see some bacteria taking the help, or the body may be taking some help from the bacteria. Because in the last week, in a meeting, I heard from one of my colleagues that in plants in desert areas, they can tolerate higher temperature up to 60 degrees because of presence of antiphytic fungi. Those fungi support the growth of the plant at higher temperature by supplying some essential enzymes which are very heat tolerant. So maybe we can think about this. This could be a new area of research, how to tame gut microbiome to make us fit for adaptation to the new climatic situations. So I have put this slide, but I don't see the person for whom I have put. In the last talk, Professor T.K. Dutta asked about gut bacteria and longevity. So anyway, this gentleman, Maximov, he's a Russian Nobel laureate. He wrote one book, The Prolongation of Life. But Nobel Prize was for different purpose, not for this one. So he observed that the Bulgarian cattle farmers, they very longer lives. And they carry their milk from the cattle farms, which are far away from their homes. And while carrying this, by the time they reach their home, they become yogurt, like curd. And they consume that way. And Maximov thought that there has to be something in this milk or the milk product that is helping their longevity. Although it is just observation, it was not a systematic study. So still I have put, maybe there's maybe something, but there are many criticism maybe there, because they also do a lot of hard work. So you cannot say alone that this particular food is helping their longevity. And subsequently, some other person has isolated one bacterium from that particular curd, and nowadays, globally, all MNCs, most of the MNCs, they use this particular strain of bacteria for production of yogurt. And another study came just last year, and I probably answered in the last seminar, but wrongly I said it's a Dutch country, but it's not Dutch, in Italy. So there is a study where they have shown that the microbiota changes with age, and in the elderly people who are of the age 105 and 109 years, the core microbiota gets shrinks, and subdominant species, they become more abundant. But how they are helping the longevity, or what is the relation, that is not known. More studies will be required to answer this question probably. Justin Sonnenburg from Stanford University, he's a pioneer in the field of gut microbiome research. So he has the opinion that synthetic biology should help designing microbes for particular diseases, or treat particular diseases or particular situations. You can expand this information to say that you can design organisms to adapt to your gut and perform the jobs that you want. And that's how you probably can have a designer gut that will be like more suitable for your purpose. And then we can make the human being more adaptable to the situations that is required. The fecal transplant is a new terminology, probably many of you have heard, that is already coming in a big way. Though the gut microbiome research is still I'll say in infancy, we don't have proper understanding. Multinationals, they are like hurrying. They are taking out many products. So fecal transplant, this particular company, OpenBiome, they have developed one kind of transplantation technology that can cure antibiotic-related diarrhea. And then they claim that there's a 90% success rate in this case. And already I know that more than 20 or 30 companies are working in this area. In India also, Vedanta, and I know a few companies who are thinking about developing some products based on human gut bacteria. But before that, we need to do a lot of work to understand the microbes in our gut. So, because if you don't understand their functionality, what do we know that Their taxonomic groups we know. Somehow, some way we know how they increase or decrease in particular situations, but we don't know exactly what are their functions for each group of the organisms. So there is a difficulty, and people say that, microbiologists say that, this is one ecosystem within our body. But how this ecosystem functions, it is very difficult to understand. Okay? Just to make you understand a little bit about ecosystem, I have taken one example, very simple example. First we understand how the system works, then we will understand ecosystem. Okay? I have taken the example of ISST. If you look, this may not be 100% accurate. This is just for example. From director up to research scholars, they have different functions. You can link this way probably. And we have then finance, and then we have administrations. Each of them have different functions. And you can say maybe the director, then finance, administration, and then heads, they may be of core committee, core group. And your research scholars or RA, they can be, or contractual employees can be considered as transient group. They can come, they can function, and they can leave. And if there is any vacancy, sometime the good performer may become a permanent member. Okay, the transient bacteria can become your permanent member also. And then, in case there is any part, this is one equilibrium. Like in equilibrium, in microbiological term, we say homeostasis. So if there is any kind of perturbation in any part of this system, that will be reflected like by disequilibrium, or we call dysbiosis. And if the system is resilient enough, we call it resilient, it can come back to its original state within a short period of time. Or if it is not resilient, it may degrade or it may not come to the original state. Okay? Now, sometime to improve the performance of the system, we may hire somebody from outside, like Horacer, who can do conditioning of the whole system, or maybe, for a particular job, you can hire a person also, like probiotics we take. And also, or consultant, like something like that. Yeah. So, like I can draw this in just 5 minutes because I know the function of each member in this group, each member of this system. And probably in this house, probably you know about this, their functions. You can also draw within a very short period of time. But let's say a person comes from outside and he has no knowledge about ISST. And in this house, we have probably people from all the groups here. And he wants to know what system is prevalent in this organization. So what he can do, he can collect biodata. If he has enough time and resources, he can collect biodata from each one. That is, we call metagenomic sequence, the whole genome sequences. And then based on that biodata, he will try to figure out who does what and how they can be related. Okay? This is one way of studying relations among the members. But in that case, there is a danger. In CB also you may lie sometime. You may find many functions which are not being active at the moment. So in genomes also you may find thousands of genes which may not be functional. So that's the problem. You may not know the functions. And then if you don't have enough time, what you can do? You can just collect the designation of each person. Just designation. And then you consult some databases or some annual report. And then you figure out like which designation does what. Like if he's a student, research scholar, he'll be doing some work. If he's an Inspire Fellow, what he'll be doing. So this way he'll link up the thing and try to find the system. But again, there is a danger. Research scholar of life science or research scholar of physical science, they will be doing different research totally. So within the same species, different bacterial strains may perform different jobs. So taxonomic diversity is still, people are using, but there is a danger like this way. So this is what we call 16S RDNA sequencing. We don't do whole sequencing, we collect the designation, and we try to figure out what are they and what they are doing. Okay, this is one way of studying the ecosystem. Now the new methods are coming where people are developing some techniques where you can scan the whole body and you can track each microbe, what they are doing in the intestine, where they settle down, where they colonize, and what function they may be doing, but proper imaging technology. So you can say that if that person is not interested in doing this, he can put on CCTV in your room, and he can observe what you are doing. So based on your activity, he can probably figure out who is doing what. And then finally, he can link up and make the ecosystem. So understanding the ecosystem is very important before we design any kind of technology on this. So, because we have limited number of members here, maybe around 100, so it is very easy to make this system. But when you have thousands of different species of organism in the ecosystem, if you try to link them based on taxonomy, you get the whole lot of connections, and it becomes a cloud. And a huge lot of data you have to deal with. So this is very, very complex system. People are still trying to understand how the microbes are related, and also there are variations in different populations. Again, within the population also, you may find some variation in microbe and gut bacterial diversity. So in ISST, we initiated some work on understanding the gut bacterial diversity of some ethnic groups. You might be aware of that. We took 15 tribal population from different states of India, from Northeast, Sikkim, Manipur, Assam, and then from Telangana. And then we compared their gut microbiota with the available data around the world. And that time there were not many data that we got for comparison. But however, what we find here, that gut microbiota of the tribes of India, they are similar to the gut microbial diversity of the people from Mongolia. And we find that particular group of bacteria called Prevotella is dominant in our population. And what was our actually main aim was to find out the core group of bacteria, those required for basic functioning of your gut. And we find that these 5 bacteria, they form the core group. They are present. Core group means they are present in every individual, irrespective of their ethnicity or their food or whatever. So they are found in all the people, and they are in nice correlation with each other. This now this work has been included in the concise general knowledge book under science and technology. And this is a little bit of appreciation for the students. Like this work is now has been included for general knowledge book. And in the last month also, when I was searching our work, I found that X-Jen, one freelance journalist, who wrote a nice article on gut microbiome research in India and also future prospect. They have nicely mentioned about our work. And we did another work where we tried to see correlation between metabolites and microbes. And this work was published very recently. And already some groups like the Black Labs, they have highlighted this work. And today only I found that this work has already been highlighted by some other groups like APBN, and then it is coming in different websites. So a little bit of just appreciation. And then actually it came in the India Science Wire. I got an email last week that they are interested to highlight this work as like one of the significant work in gut microbiome research in India. So I gave them all the details, and they have already published. Now, apart from this, what makes me a little bit more satisfied is that I got an email from the Justin Sonnenburg group from Stanford asking for our data. Like they want to see our data and they want to do some comparisons, and they think our work was good. So this is like some kind of good thing about the students. And we have initiated another big research along with another 5 organizations. ISST is also a member with NCCS Pune, and then AIMS, then SRM University, NIPGM Calcutta, and IBHD. So in this project, we will be studying 90 communities of India, covering all the major ethnic groups. Similar work that we have done before. And this is our group. And thank you all.